All right, welcome to video number 5, I believe. Let's hurry up and get through this one. I've only got 6% battery left on my computer, so we gotta quickly go through this stuff. All right, so if we know the genetic makeups of the parents, what type of offspring might they produce? This is the first question that we were really trying to work on and answer using genetics. You've already done a little bit of this stuff. We just have to get it slightly more formalized. So what is the probability of producing different types of offspring? And remember, probability is the likelihood that a particular event will occur. So what is the chance that this thing is going to happen? So what are they going to look like, and what is the likelihood that they're going to end up looking that way? And the tool that we use in biology to do this is a Punnett square. So a Punnett square looks just like this. A Punnett square is a diagram showing the allele combinations that might result from the form of a genetic cross between two parents. So what's going to happen when you fertilize every possible egg with every possible sperm? The alleles of the first parent go across the top of the square. So here we'll put a big T and a little t. And the alleles of the second parents go along that left-hand side. Again, we'll put a big T and a big T this time. The possible gene combinations are going to be in the middle here. The way that I like to do this is I like to use the drag and drop method, where I'm going to take that big T and I'm going to drag it into each of these boxes, take the big T and drag it into these boxes, the big T and drop it down into these two, and the little t and drop it down into these two. The letters represent the alleles. A capital letter represents a dominant allele. A lowercase represents a recessive allele. You do already have that. This is just a little bit of review. So the first box, we're taking this egg and we're fertilizing it with this sperm. And then we're just going to go ahead and do the rest. Now, it doesn't matter if this big T comes before the little t or the little t comes before the big T. Really, it just matters that it's heterozygous here in this box. Generally, we put the dominant before the recessive, but it does not have to be that way. So, Mendel began his experiments using true breeding plants. So as soon as he discovered that a tall trait was dominant over the dwarf trait, cross a true breeding tall pea plant with a true breeding short pea plant. So our genotype for our first parent, take just a second, think about it, write it down. The genotype of the first parent is homozygous dominant, big T, big T. And the second parent, take just a second and think about it. Is little t, little t. So place the alleles of the first parent on the top of the square. And the alleles for the second parent on the left of the square. Now just fill in these squares to show all the possible combinations that the offspring might inherit. Go ahead, pause the video now if you need to while you go through and fill out this problem. Alright, you should have these filled out by now. So we're just going to go through, and each one's going to get a big T from the first parent and a little t from the second parent. Now, go ahead, fill out this table to show all of the genotypes and phenotypes. Remember, your genotype is that homozygous, heterozygous stuff we talked about. And the phenotype is what you can actually point to. What does it look like? Go ahead, pause the video now, and fill out the possible genotypes and phenotypes for all of these. Alright, you should have these filled out by now. So 4 out of 4 of them are going to be heterozygous, and 4 out of 4 of them are going to be tall. So in the above problem, none of them showed that dwarf trait. They were all tall. As we learned earlier, he looked and wondered what's going to happen if he allows that F1 generation to self-pollinate. So take just a sec, think about what your Punnett square is going to look like if you allow those to self-pollinate. So by taking 2 members of that F1 generation and crossing them. Alright. So the genotype of each parent are going to be the same. So they are both going to be big T, little t, big T, little t. We take that first parent, we put it across the top. Take that second parent, put it across the side. Now, take just a minute, pause the video if you need to, and go through and solve this out. Alright. So in our first box, we have homozygous dominance. Second, we have heterozygous. The third, we have heterozygous. And the fourth, we have homozygous recessive. This is the problem that we did in class with our 16 bubbles. So our genotypes, 1/4 homozygous dominant, 1/2 or 2/4 heterozygous, and 1/4 homozygous recessive. With these, anything that's going to see that big T, that tall allele, is going to be tall. So we have 3/4 tall for our phenotype and 1/4 short. The 1/4 short only appears because both of these are the heterozygous trait. So having dimples is dominant over the absence of dimples. Cross a heterozygous dimpled man with a woman who does not have dimples. Show all of the work in the Punnett square and summarize your findings in the table. Go through, work out this problem for me, and I'm going to see you in the next slide.